All right, ladies and gentlemen, welcome back. So at this point, we have talked through the first two major steps of cellular respiration, glycolysis, and the citric acid cycle. Today, we're going to pick up with that final major component that is oxidative phosphorylation. And so what we're going to see when we look at oxidative phosphorylation, this final component, we're actually going to break it down into two subphases, and that is your electron transport chain as well as then chemoosmosis. So oxidative phosphorylation is those two components put together, and it's really what's going to drive the bulk of the ATP synthesis in cellular respiration. It's this process here that gets the bulk of that ATP produced in the vast majority of organisms. So what it really is doing is taking all of those electron carriers that we produced during glycolysis and the citric acid cycle. So all of that NADH and that FADH2 that we've been slowly creating by pulling glucose apart, pulling off carbons, manipulating and moving around energy from electrons and hydrogens. And now we're going to take those electron carriers, they're going to donate electrons to our electron transport chain, as well as yank off those hydrogen ions, those H plus ions. We're going to shove them into a concentration gradient, and that's going to allow us to then power ATP synthesis via this oxidative phosphorylation. So how does it work? Well, your electron transport chain is sitting here. It's a bunch of proteins that are sitting here in the inner membrane, embedded in that inner membrane of your mitochondria. And all of those cristae, all of those folds in those mitochondria are huge for being able to increase that surface area, which means we get to increase the efficiency of the transport that these proteins are going to do for us. Um, so these proteins, remember, are going to allow us to break down that energy, that free energy that we're getting from these electron carriers that ultimately had been bound up in the bonds in glucose, and do it in bite-sized chunks, right? So that we can do it a little bit by little bit by little bit, so that we can actually harness that energy and store it. Otherwise, we'd release way too much energy at once, and your body would spontaneously combust as a result. The amount of heat and energy coming out of that wouldn't be something that your body could handle. So, this chain allows us to do it in smaller steps to release that energy in manageable amounts so that we can actually tap into it and use it. So, when we look at the enzymes here, these protein complexes, their job is going to be to yank off the electrons that are going to then get fed down this chain, as well as pull off those H pluses and create a hydrogen gradient that's then going to be able to allow us to drive that chemoosmosis. So ultimately, the electrons are going to go down this chain. They're going to end up being tied back to those hydrogens, as well as then getting tied back to oxygen. So oxygen is going to work as our final electron acceptor here to ultimately create water, which then comes off as a byproduct. So we look at the steps of this electron transport chain, each protein is going to pump H pluses from that mitochondrial matrix, right, which is the most internal component, sort of the jelly-like substance inside your mitochondria, and pump them into that inner membrane space, right, the space between your inner and your outer membrane. That's going to result in this H plus gradient. And keep in mind that we need to yank off those electrons from that hydrogen to make a hydrogen ion in order to keep them in that inner membrane space, right? So it's a phospholipid membrane, which means certain things are able to pass through it easily and certain things aren't. We want these to be H pluses. We want these to be hydrogen ions because we want them to stay between those two membranes. We don't want them diffusing naturally back through. So we're going to pull off those electrons from those hydrogens, use them as energy to make the chain pump work. We're doing active transport here, but instead of using ATP, we're using the electron transfers from those NADHs and FADH2s to make the pumps work. And then we're using those hydrogen ions that will get stuck there because they are by definition ionic, so they are hydrophilic, so they won't pass directly back through the membrane to then drive the second piece, which is chemoosmosis, which is going to be a passive transport system. So we're really coupling an active pumping system here with a passive transport system in the next subphase of oxidative phosphorylation. Interesting thing about these protein complexes is that they are multi-proteins, so it's multiple proteins in a single complex that I'm showing you here, right? So there are multiple proteins going on here, multiple proteins going on here. The other interesting thing is that they are proteins that include cytochromes. Basically, they're iron-containing proteins. Um, so if you know anything about the condition of being iron deficient, so anemic, right? If you have anemia, that means you don't have enough iron in your body, and that has a range of effects. Um, you're tired, you're fatigued easier, there's a whole host of reasons why. One of which is the hemoglobin in your body needs the iron to more efficiently bind oxygen to get it from your lungs to your cells. You need the oxygen in order to do this process, to be that final electron acceptor, but you also need the iron in order to make these protein complexes work efficiently at yanking off those electrons and making that hydrogen gradient, which ultimately makes ATP, which is your energy. Um, and so being anemic has a compounding problem here. You can't get the oxygen to your cells as efficiently, and even when you can, the amount of ATP you're able to produce as a result is also lower because your electron transport chain isn't doing its job as effectively because you need the iron in the proteins in the electron transport chain. So it's just kind of an interesting sort of side note there. So, like I said, we're going to create this H plus gradient, right? So here is your mitochondrial matrix. Here's that inner membrane with all of those folds. Here's that inner membrane space. And what they're not showing is the outer membrane out here. So we've used that electron transport chain to force a concentration gradient of H pluses, right? We've used that active transport to make this gradient and keep it in place. What chemoosmosis is going to do now is couple that proton motive force that we have created, and we're going to allow these H pluses, the only place they're going to be able to diffuse back into the mitochondrial matrix, so go from where they're high to where they're low, is we're going to force it through this ATP synthase, this um, enzyme protein complex that's sitting in this membrane. And what this does is kind of work like a rotor, if you will. So every time an H plus comes through here, it causes the complexes in here to turn, and that's going to basically snap together the phosphate plus an ADP and churn out the ATP. So every time a hydrogen comes through here, this turns, these guys get smashed together, and we make ATP as a result. Way more efficient than that substrate level phosphorylation that we've seen thus far. So this is still an enzyme, but because we're using the ability for those hydrogen ions to come through here and force the additional energy component to make this enzyme work, it's way more efficient than the substrate level we've seen thus far, where we've used some other substrate that had the phosphate attached, had the ADP in there, and we had to yank off the phosphate and then attach it. Or getting a much more efficient ATP production here. So again, if we zoom back out and look at this as a bigger picture, here's your electron transport chain, right? Using those NADHs, those FADH2s, yanking off those electrons so we can keep these pumps working, moving those H pluses here into that inner membrane space. When they come back through, we'll take some of those hydrogens that are now here, take the electrons that are coming down this chain, attach them to those hydrogens and that oxygen, produce water as a byproduct. The H pluses then come through that ATP synthase, help it to rotate, and then allow us to be able to add that phosphate back to that ADP, create the ATP as a result. So we're taking this energetic flow of H pluses to drive the phosphorylation of that ATP. Okay, so let's zoom back out even further. So if I look at the major inputs and outputs of each of these processes, where do I get all the energy from? So I go through glycolysis, right? I take that glucose, I break it down into 2-pyruvates, I get some NADH off as a result. Those 2-pyruvates get converted into acetyl-CoA, I get a little NADH off from there as well. Shuffle that acetyl-CoA into the citric acid cycle, I get even more NADH and FADH2, which goes to my electron transport chain and chemoosmosis. Glycolysis, right, I get to net 2 ATPs via the substrate level phosphorylation. Citric acid cycle, I get to net 2 ATPs again from substrate level phosphorylation, so not particularly efficient there, but I get a little bit of energy out as a result. And then I go through oxidative phosphorylation, and I get somewhere on the order of about 32 to 34 ATPs. That number there at the end can range. Sometimes it's even lower, right? And we can talk more like 26, 28. Sometimes you get a really efficient system, and you may see up to even as much as 40 ATPs. So there's a range here. Reason being, it's a whole host of things. Um, how efficient is that system? How quickly is it working at that particular time? How much iron is present can have an impact on the number of ATPs. And it can vary from one moment to the next. It can vary from one cell in a multicellular organism to another cell. So there's a range, is my point. So when we look at the collective total, most textbooks will talk about somewhere on the order of about 36 to 38 ATPs is the pretty consistent general average for most organisms in most cells. But if you look at that and we go back to the amount of free energy that's stored in those glucose bonds versus the amount of energy we actually get out of it, it's only about 40% effective, or 40% efficient, which doesn't sound spectacular until we start talking about fermentation in a little while, and you're going to see just how ineffective or inefficient that really is.